Thank you, everyone, for to be here in almost the last talk today, and thank you for the organization for the invitation to participate in this event. And also because it's my first time in Prague, so I'm more than happy for that. And thank you also for the speakers and interesting talks and questions too. Now in my presentation, I focus on the genetics applied to the human reproduction. This is my declaration of interest that I have no any conflict, and this presentation is purely academic. Well, this is the basic dogma of biology. This, our cells have DNA, is the instruction book that have the information based on the combination of 4 letters, A, T, C, and G, and have the information to how we are. There are more than 50,000 genes. Gene is like the chapter of this instruction book. And this information is transcribed in mRNA that is translated to proteins. Proteins, which have a different function, that define our phenotype. Understanding phenotype not only is we are tall, fat, or brown, also how a nutrient can be assimilated or a drug can be affected. There is a different point in the history that is very important. For me, the development of the new technologies is the most important, because we have now more information about the different science, like the genomics, like the transcriptomics and proteomics. The application of the different science in the reproduction area is called like reproductomics. Reproductomics is we can study with these different science different stage in the travel of the infertility patients. In this talk, we have seen the different analysis, the different study that we can do in different stages. Preconceptional, previous to obtain the embryo. Preimplantational, when we have the embryo, we can study it. And also prenatal, when we have the pregnancy and go on. Well, we had started with the preconceptional stage. Then we have a couple of tests, a couple of studies that we had to do all the parentals and also to the donors. And they are very important, is the conventional karyotype and the carrier screening test. The karyotype is the photograph of the different chromosome. The chromosome is the structure of the DNA. Humans have 22 pair of chromosomes and also the sexual chromosomes. In the karyotype, we can see this chromosome ordered by size. No? The first chromosome is the larger, and the 22 chromosome is the smaller. Missed you. Well, this karyotype show you information about the number of the chromosome, but also the structure of the chromosome. And this is important in reproduction, because it could be the cause of the repetitive miscarriage and also implantation failures. Because if you have all the chromosome, you are a phenotype normal, but you modify or you have changes in the number of the chromosome, you show a pathophysiology. But a normal karyotype that includes, well, here you have a image, a picture about a normal karyotype. This is a male, because it has a X and Y chromosome, and they have 2 copies of each chromosome. This is a normal karyotype. In this picture, you have a alterated karyotype. This is a woman. They had 2 chromosome X, but you can see that the 22 chromosome, you have only 1. But it's not missing. It's here attached to the chromosome 15. This is a normal patient. The phenotype of this woman is normal. But when she generates the eggs, the gametes, could have problems with this embryo combination. So the study of the karyotype is a test, is a study that can be actionable. If we identify a couple with problem in the karyotype, we can use the PGT-A that we talk later to select the healthy embryo. No? So it's a study, a basic study that we have to do to all the patient, and also very important to the donors to include or not in the bank. The other test we have to do for all of them is the carrier screening test. Well, all of us, we have 2 copies of our DNA, one from our parents. The carrier screening is based that the DNA, the letters of the DNA is similar in the human. The 99.9% of the DNA is identical in the human, but there is modification, mutation that changes in this letter that do that we have different. This mutation explains also the disease. When we have a mutation, a dominant mutation in only 1 copy of our DNA, we demonstrate the phenotype of the disease. But there is the other kind of disease, that is the recessive genetic disease, that you require mutation in both copies of your DNA to demonstrate the illness. But if you have only 1 mutation in one of these copy of DNA, you are carrier of the illness, but no demonstrate the pathophysiology. So all of us are mutants. All of us as carrier or of a recessive disease, but we don't know because we haven't a demonstration of the disease. The problem is when our couple of the donor have the same have mutation in the same genes, because they can generate a child with rare disease. The rare disease are rare because the number of this illness is very low, but they are more than 7,000 of disease. So the combination is very important. Also, the characteristics of the disease is that the diagnosis is very difficult, the delay in the diagnosis, no treatment for this kind of disease, and the best option is to avoid that child born with this kind of disease. And this kind of test, the carrier screening test, show the carrier status of this kind of disease. We evaluate by sequencing the presence of the SNPs, that has a single nucleotide polymorphisms, changes, this is mutation in this letter, and also the CMV, that are segment of DNA that can be duplicated or deleted. The detection of this mutation, this alteration, can evaluate the matching between the couple of the person with the donor. The clinical results show that most of us have mutation, are carrier of this kind of disease, but this point is very important. 2.7% of couples are carriers of the same disease. One of 40 couples are very risk to have children with this kind of disease. So we have to evaluate, to communicate to our patient this risk, and we have a tool to measure the risk. And I said with the karyotype, if we identify this risk, these kind of steps are actionable. We can act to avoid the birth of this disease children. Also, the society recommends this test, so we had to do because it's improve the probability to have healthy children. We developed a tool that is called HERES, is determining implantational stage. We can use other implantational stage. We can use other tools to study the embryo, like PGT-A, PGT-M, PGT-SR, and also the non-invasive PGT-A. But the indication of the PGT-A depends on the patients. PGT-M is indicated when couples carrying or affected by monogenic gene disease. PGT-SR is indicated when the couple carrying chromosomal structural rearrangement, we say in the karyotype, and PGT-A is indicated for advanced maternal age, recurrent implantation failure, severe male factor, recurrent miscarriage, and also conception with a previous anonymity. And it's important to understand the legislation that depends on each country to perform this kind of test. In the case of PGT-M, how we say, is indicated when we have a specific genetic cause of this kind of disease. Technology have improved in the last year, and now we use NGS to identify this mutation in the embryo. It's important that we have a previous studies to do in the parental. Sometimes we have to do segregation study to identify that mutation. After that, we study the embryo biopsy to select the best embryo to transfer. In the case of PGT-SR, we say is indicated when we have chromosomal structure rearrangement, and the technology has improved also in the last year, and now we are using NGS to evaluate these changes. Changes that affect more than we thought, so it's important to identify this change in the karyotype of the parents. Here, in the operation of the PGT-SR, we also have to do previous pretest studies in the parents. And the PGT-A, that is the most used of the preimplantational test, is indicated for a different situation, but the most of the cases are related with advanced maternal age. It's important that only 22%, this is SARF data, of IVF cycle produce a pregnancy, but how do you know, this depends on the range of the age of the mother, because the pregnancy decreases with the age if we don't study the embryo. And the implantation failure is increasing with the age because the anniversary of the embryo is increasing. The technology for PGT-A is also the NGS, and here we have to do the test in the parental, but it's recommended the genetic counseling previous to perform the PGT-A to the embryo.
The change of the technology allow us now with the NGS to have a high resolution, identify segmental aneuploidy, polyploidy, but also we have now a concept that is the mosaicism in the embryo. This is the picture, the normal picture of we have after the NGS analysis. We have confidence range that in the middle you can see the amplification of the different embryos. If you have inside of the confidence range, this embryos is considered euploid. When we have an amplification of a chromosome outside of the range of the confidence range, we identify an euploid, trisomy of euploid. And sometimes this embryo have a complex abnormal. They have a lot of aneuploidies. Literature showed that the clinical implantation rate and sustained implantation rate improve with the study of the embryo by PGT-A. With the age, in the cases that we exclude in the PGT study, we observed that with the maternal age, decrease the live birth, but increase the miscarriage. But when you analyze, when you select the embryo with PGT, the live birth have a similar level independent of the maternal age. The mosaicism is defined like the presence of two or more genetically distinct cell lineage in an embryo. In the picture that we we saw previously, we talk about the confidence range. In a mosaicism, we detect a strength amplification that is not in the intermediate. So we can identify with this technology the presence of different lineage. In this internal data from Data Science, we have an incidence of 11% of mosaic diploides in the embryo that we studied. And this percentage is independent of the maternal age. Attending to different points, we can classify the embryo in base of the mosaicism. Attending on the number of aneuploidies, simple, complex, the type of aneuploidies, it affects the Y chromosome or all the segmental, and of course the level of mosaicism, low range or high range. Different picture to identify this different classification of the mosaicisms. And when we have a normal blastocyst with the high implantational potential that they have, the low rate of miscarriage and increasing ongoing pregnancy, we advise the transfer of this blastocyte. When we have abnormal blastocyte, we not recommend this transfer because they have no implantation, high rate of miscarriage, or being disaffected. But what's happening with mosaicism when we detect mosaicism? Well, the clinical result showed that the implantation rate, the ongoing pregnancy rate, and also the abortion is affected in the different mosaicism embryo compared with euploid embryo. But the newborn outcome doesn't show this change. So independently, we transfer euploid or mosaicism, specific mosaicism embryo, the weight at birth is similar in the different group, and also the level of gestation. So it's important this kind of data because show that the mosaicism embryo with mosaicism can reach a birth in a healthy birth. This kind of mosaicism have ability to implant, but they have higher miscarriage, but finally they have birth of healthy children. The PGT's position with the transfer of this embryo is here they show different recommendations that we suggest in the report of PGT. And the non-invasive PGT-A that we talked in the previously is based on the analysis of the shed DNA into the sperm media, that is when the embryos is growing. The advantage of this technique is that we can evaluate the karyotype of the embryo without the potential for damage from biopsy. But these cells that we see in the media have different sources, and they have a characteristic that we have to take in account. This is the variability in the amplification range. We have higher range of ploidy concordance. It depends of the depth of culture and also the size of the culture drop. So it's a new technology. We have obtaining data, but we have to take in account this limitation of the technique. Today, the biopsy of the embryo is the gold standard to study the PGT, but this new technology have other challenges like the detection of mosaics and segmentals. After talk about the different technologies study that we can do in the embryo PGT in the different ways, we can study also in the pregnancy the karyotype of the baby by a non-invasive way, by the NIPT. NIPT is based on the C-free DNA that we find in the maternal blood, and we can see the trisomy and also the microdeletion by NGS from this material. All these tests is applied to patients with low complexity. But what happened with high complexity cases? In the cases of the implantation failure or repeated miscarriage, we had to study again at the preconceptional stage. And here we have different study, different analysis that we can do. It's important that we have a miscarriage, the study of POC and thrombophilia. The POC is the proof of concept. We can study the karyotype of the abortion. The aim of this study is identify if the cause of the abortion is the embryo. This is the embryo, but we know that in the next cycle we have to use PGT-A to avoid a new abortion, a new miscarriage. But if the embryo is okay, we have to find other cause. Other possible cause is the thrombophilia. Thrombophilia is a clot in the vessel that increase the vessel pressure and is finally provokes the miscarriage. Thrombophilia could be congenital. In our DNA, we can identify if we have a higher risk to have an event, a thrombophilic event, or acquired. Identify this risk allow to it's an actionable test. We have the possibility to treat the patient with heparin, low molecular weight heparin, to prevent this situation. But it's very important, personalize the treatment. we show them to treat in the same way of the patient. We had to treat with low molecular weight heparin in the cases that we identify congenital or acquired risk for.
I'm sorry, may I push you a little bit? I have 5 minutes
Okay.
more. Do you think it's okay for you? But
Yeah,
we have
it's
also
okay.
time for questions. Thank you.
No worries.
Thank you.
Patient with a real risk for thrombophilia. We have tests where we identify the different mutations and evaluate the risk of the patients. And of course, we have to study the endometrial. Endometrial, the embryo evaluation, we have different techniques to study the embryo, but you know that the application of any of these techniques is not really able to improve the quality of the embryo or the possibility of its implantation. It only allows us to select the best option. The implantation is the process where an embryo attached to this tissue, the endometrium. Endometrium is characterized by receptivity and also functionality. And there is a molecular dialogue between both the embryo and the endometrial tissue to be synchronized. Well, it's important the physiology of the endometrium that is very vascularized. It's the only tissue with the capacity of generate new vessels in its cycle. This new vascularization is controlled by the immune system. The receptivity is this time, this period of time when the endometrium accept the implantation, accept the attach of the embryo. In natural cycle, it has between days 19 and 21. With progesterone, we know that the most of the women are in receptive after 6 days of the treatment. It's possible that the window of implantation could be displaced, and this kind of test can help us to select the best moment to transfer a healthy embryo. We identify if it's receptive, pre or post-receptive, and personalize the moment of the transfer. We know that currently there is a controversy about this kind of test, but I remember the personalized medicine. Not all the tests are for all people. We have to select which patient could be a benefit with this kind of with this test. And we talk also for the functionality of the endometrium that is very vascularized, and the microbiota and microbiome affect the immunologic system and control this vascularization, the new generation of vessels. The microbiota is referred to the community of living microorganisms, but the microbiome is also the microorganism, but their genome and the metabolite that they generate with its activity. Now we know that we have billions of friends that we didn't know we had, and we have changed our vision of the implantation procedure. We have the endometrium with different cells and the blastocyst, but now we know that there are many microorganisms that affect the functionality of the endometrium. We can evaluate the presence of the different bacteria, viruses, fungi, and if we identify pathogens, we can treat them. And also we evaluate different metabolites to understand the situation of the immunological system of the endometrium. We have created evidence, we are presenting the different works about the role of the microorganisms in the endometrium, and also the candida that affect not the implantation, but is affecting to the miscarriage in this kind of patient. And what should we do with them? Because we studied the woman, the endometrial, but we don't study in many cases to the other part of the color. But then in the literature, we didn't observe works that talk about the plasma infection that can affect in 4 ways to the male infertility, adhering to spermatozoa, production of rods, cross-reactivity antibodies that recognize the surface of the sperm and reduce the nucleotide activity and impedes the ability of the sperm to penetrate the egg, and also affect to the pregnancy work. The other test that relate the microbiome with different parameters of the sperm, like volume, pH, viscosity, concentration. We have a test that evaluate, analyze the ejaculation microbiome. Finally, in conclusion, we have different different tests that we can use for our patient, but it's important to personalize, to select the